Hello and welcome to the DDW Meet the Researcher Vodcast, a series of in-person interviews. I'm Megan Thomas, Multimedia Editor at DDW. At the SLAS 2024 International Conference and Exhibition in Boston, which took place in February 2024, I met with Nicola McCarthy, Head of Research at the Milner Therapeutics Institute. Thank you so much for joining me today. Could you start by giving me your name, your job title, what you do?
Yeah, sure. So I'm Nicola McCarthy. I'm Head of Research at the Milner Therapeutics Institute, which is part of the University of Cambridge, and I oversee both wet and dry labs at the Institute.
Great, thank you. So the first question I have after that is, can you provide a top-line summary of your research?
Sure. So what we're interested in at the Milner is basically partnering between pharma and academia. So we have a big consortium agreement that at the moment involves 14 pharma companies. And then we work to partner them with academics working at the University of Cambridge, Babraham Institute and the Sanger to see where they've got coalescing ideas, coalescing interests. And it's a way of pharma being able to dip their toes in new areas and de-risking from that point of view. From the groups that I actually oversee, we're developing, in the wet labs, we're developing new models for arrayed CRISPR screening. So we're looking to work with primarily human material as much as we possibly can. And then in the dry labs, which is headed up by a bioinformatician, we're looking at big data sets, patient-derived RNA-seq data sets, for example, to see whether they can be mined to try and identify new potential targets.
Great, thank you. That sets it up very nicely. The next question is, where do you think the biggest opportunities are in this field?
I think it's moving into primary tissue. So certainly for the wet lab work that we do on the arrayed functional genomics, it's very much a case of moving away from the cell line models, moving away from two-dimensional models, moving into complex co-cultures with human tissue or organoids, for example, patient-derived or iPSC-derived, and working out ways of using all of the information that's been gathered from working in the cancer cell lines to see whether you can make those models scalable and robust enough to translate them into the pharma pipeline. So that's kind of where we're working with that. And I think, again, the ability to bring deep machine learning and AI into the big data sets is one of the areas.
Great, thank you. So what are the challenges then in taking this to market?
I think really it's the scalability and the robustness in terms of the wet lab work. So it's trying to get those models so that they can at least handle a substantial gene knockout set. And that for every biological replicate, you get enough reproducibility between well by well and plate by plate. I think for those models, those are the real areas that we need to solve at the moment. In terms of the big data from the dry lab side, I think it's really coming up with the right training sets and the right way to address biological questions or patient derived questions such as, can you identify patients that are going to respond to drug X, drug Y? I think we've made a really good start in that area, but I think there's a lot more work to do.
Yes, well, hopefully everybody gets behind that and agrees. This is possibly a hard question. What has been the highlight of your career so far?
I think it's variety. So I've worked in academia, publishing, biotech, and then back into academia. And I think really meeting lots of different people with lots of different ideas and being able to learn throughout my career, no matter what I'm doing, I think that's always been the major highlight for me.
It does seem to be lots of variety in this sort of field, so yeah. That's good to hear. And which industry-wide drug discovery breakthrough has been most impactful to you in this time?
I think probably it's both the big data. So when I was working at Nature Reviews Cancer, we saw a lot of the development of the big deep learning to sift through all of the omics data that was coming out in the cancer research field. I think in terms of wet lab, really for me in the last 10 years, it's really been CRISPR. So that's been the big mover, really.
That does seem to be a consensus across many people.
Yeah.
So what is the best piece of career advice you've received?
I think it's always do what you love, what you have a passion for, because you're going to be doing it for a long time, so you need to enjoy it.
Yeah. And it shows in the work that you end up doing.
Yeah,
you're passionate
absolutely.
about it. And what advice would you offer someone looking to follow in your footsteps?
I think it's not allow yourself to be pigeonholed. I think that anybody with a scientific background, or even any background that they've got a deep knowledge in, you pick up all sorts of different tools and you can apply them anywhere. So really you can work wherever you choose to work, and you shouldn't let people say, well, no, you're not quite right for this.
Yeah, a bit of a follow-on from that is, do you think that's gotten better in kind of recent years, or is it still an issue?
No, I think it's got better.
Okay.
I think when I was first working, it was very much a case of you were either an academic or you worked in industry, and never the twain shall meet. Yeah. I think that's changed massively now.
Thank you. And now the last question I have for you is, if you could make everyone read one book, article, academic paper in this field, what would it be and why?
That was one of the questions I felt really, really difficult to answer, because I think things that inspire you were very individual to your journey. Mm-hmm. So I would just say that it's just to remember the papers that you read, really maybe when you were an undergraduate, and thought, wow, you know, that's really fascinating. For me, it was a lot of the work of Paul Nurse and the yeast biology work that he did. And reading Richard Dawkins' Selfish Gene, again, I think was inspirational to me. But I think it's remembering that passion that was ignited at that point.
Yes, and that links very nicely to your advice to keep passionate.
Yeah.
Remember the roots.
Yeah, absolutely. Yeah, especially when you get those really bad months where nothing's working, which we've all been through. Yeah.
Yes. Well, thank you so much, Nicola. That's all the questions I have for you. Enjoy the rest of SLAS, and thank you for joining me.
Thank you very much.